No? Let's start. No. So good afternoon, everyone. Thanks for coming. I think a few of us are also getting our food, but we'll get started. And I'll try to be as lucid as possible in my explanation. I am known to ramble on. So if you think you're getting sleepy, please raise your hand so that I'll speed up, right? So, but the main goal over here is to actually tell you what AFA is, which stands for Adaptive Focused Acoustics. There won't be a quiz after this, so don't worry about me asking the full form of AFA, as long as you remember AFA. So Adaptive Focused Acoustics and working with FFPE samples. So yeah, he likes to exaggerate. So any show of hands, how many people over here are working with FFPE now? Anybody? Nobody's working with FFPE, okay? So we all know what FFPE is, right? So I'll go over that. But let's get into what we are going to do today. So first and foremost, how many of you, I know at least one, but how many of you are aware of AFA or this technology? 1, 2, okay. The less, the better, because I then, my slides are then justified, right? So I'm going to introduce AFA and highlight why this technology is better than what is typically used by most people. One other question, how many of you are actually working with DNA, RNA versus how many working with proteins? No, DNA, RNA? Oh, good number. And proteins, anybody? You're working with proteins. Awesome. I love proteins, both on my plate as well as in the lab, it works. So, we're going to go and discuss, why AFA? What's the value of AFA? Why should one use it? For a lot of reasons. Then we'll get into introduction of FFPE, which I think is going to be a new one for all of you, and show some examples and so on and so forth. So adaptive focused acoustics basically is a way, it's an ultrasonication technology, but what it does is basically it sends acoustic energy at a certain frequency. And this frequency is sub-bass, or somewhere around 1 megahertz frequency. Now, the first thing that might come to your mind is that how and why is it so different from typical sonication? And the answer lies to the right of the screen, that typically what sonication does is basically it's uncontrolled energy. So you put your samples in a bat sonicator or you stick a probe sonicator, and first of all, forget the contamination risks. Let's move that aside. Even if you ensure very well that there are no contamination, even then, when you get to that point, it generates a lot of heat. Most importantly, when you're using something like a bead beating, like you put beads and you shake it, and there's a lot of heat generated. What does that mean to your unstable samples? Even more so when you're working with proteins or RNA, which are not very stable. They can disintegrate. You can basically jeopardize your analytes. So this is the technology that acoustic energy is delivered to the sample at a certain frequency without touching the sample. So it is delivered via a magnetic transducer, which is sitting in a water bath. Now, these are delivered to your sample, which is in a certain well plate or a tube, and they have some amount of impurities in the plastic or glass, which causes a lot of cavitation bubbles in the sample. Over a period of time, these bubbles collide, just what you would see in the shower. A lot of droplets, they eventually move, and they move to form a bigger bubble, and then it rolls down. So here they implode, generating some amount of energy, which is the shearing energy that is used for disrupting tissues, homogenizing cell lysates, working with whole blood cells, working with any kinds of tissues, so to speak, organoids, and so on and so forth, and then FFPE. So it is maintaining at a certain temperature, so it's temperature controlled. You can keep it at the temperature that you desire. We typically work with 7 degrees, 10 degrees Celsius, so it works in that temperature domain. So no heat imparted to the sample and no contact with the sample. So over the years, this has been, I mean, this is the energy difference I was referring to. And this has been used, so this company, Covaris, and this technology has been around for decades now. So we are 23 years old now as a company, and we are almost considered to be the gold standard for DNA shearing when it comes to next-gen sequencing labs. So most of the next-generation sequencing labs that are working with tumor tissues or cells, looking at whether they're cancerous or not, they look at the genomic or genomic profiling. So they look at DNA and they want to have a DNA of a specific size. So for those who are not in this space, they would, it's for them. Those who are already working in this space, I'm preaching to the core. But you reduce your DNA down to a certain specific size, which is called DNA shearing. And then you amplify it and you find your sequence and match it with the library to see if it is cancerous or not. So that whole thing, we are considered to be the gold standard for sample prep. Illumina sequencers and their workflows name our instruments to be used as a must-have, so to speak. So it goes much beyond that. It's not just for DNA shearing. It is not just for RNA or RNA shearing, if you may. It's also for proteins. It can also work for metabolites. It can work for a host of tissues, a host of sample types, matrices, as I mentioned. So I will come to this slide again later in the presentation. A lot of it is pertaining to things like proteins as well, especially the purification cleaning part. I'll elaborate on it as and when we get to the protein part. But one thing I would like to draw your attention to, if you look closely, the rotated Wi-Fi signal, can you all see that? So that's kind of our logo for the AFA. And it's actually the inverted Wi-Fi is how we see it, because the epicenter is at the bottom and it's radiating up top with the energy, right? So this rotated Wi-Fi signal, it's changing sizes, right? And what does that mean? It means that while you are delivering this energy at a certain frequency, which is constant, and this frequency is a long-range frequency, as I mentioned, it's sub-bass. It's like, for those of you who are into music, you would know that the bass always hits the wall because these are long frequencies. And hence, mu is equal to C over lambda, so it's inversely proportional to the wavelength. Hence, it's shorter wavelength. What does that mean? That means with short wavelength, you have a short stick in hand. You can point at something with much more ease. Hence, the delivery of the energy is much more focused and on the sample. So that you do not cook the sample, you do not heat other parts of the sample or the tube or the plate if you don't wish to. So that's one advantage. But another thing is you can change the power. So what does that mean? That sometimes you need a lot of power to go through certain types of tissues, let's say skin, brain cubes, and sometimes you don't need as much power. It works with much less power. For homogenization, you don't need a lot of power. So those are the times where you would need different power settings, and that's where you actually have the ability to customize. And there are several other customization points that I will address. So over the years, as I have been saying, we have seen this technology being used extensively across different kinds of applications, whether you take metabolomics, proteomics, biotherapeutics, which is biopharma, so to speak. Those I started with them because those are my favorite topics. And then, of course, in next-gen sequencing, in DNA, in RNA, so on and so forth, and for biomarker extraction. So, you name it, we have seen whenever people had difficult matrices to work with, it actually, AFA came and helped generate a lot more analyte than needed in a higher concentration, higher quality. So I mentioned the 10 reasons, and we do not have to go through all the 10 reasons, but one thing, for those of you who are working with DNA, RNA, and DNA shearing, you would know that there is some significant issues when it comes to shearing them in terms of the bias. So this is a technology, because it's external, it's energy driven, there's no GC bias. So when you shear the DNA, it is an unbiased shearing, which is why you will also have a much tighter shearing. So if your target is, let's say, 175 base pairs or 250 base pairs or whatever, you will have a much tighter grouping. You will not have a variability that's like for 175 base pair target. It will not be like 125 to 250. You will probably see it between 165 to 180. That's the range with tighter grouping, which means you have better yield of your target DNA. Hence, you will have better data or better confidence in the data downstream. So we can continue to do go over some of the other things. It is, first of all, it's non-contact, so you're not touching your sample, no contamination, and I mentioned narrow distribution. It becomes a part of every workflow. So whatever your workflow is, AFA sits at the bottom, so you are not having to change your workflow, so on and so forth. So I initially thought that this would be a good t-shirt, but it's too much of text to be converted into a t-shirt. It will probably become more of a bed sheet by the time we get to it. So I avoided, I put it on all the slides. So as I mentioned, it's the gold standard for DNA shearing. We are seeing this being used for multi-omics as well. So metabolomics, proteomics, we are seeing that's being used. And for you in particular, who is in the protein world, you probably have heard of names like Matthias Mann, Jenny Van Eyck, Neil Kelleher. They're all using these instruments. So they're all, John Yates, most of them are using this instrument in particular. ASMS, this year we actually, we had a lot of people come to us, come to our breakfast session, so on and so forth. It is scalable. So whether you're working with one sample at a time or 96 or 384, we have instruments that can cater to all those productivity throughput requirements. And the last part is the direction where we are heading now is going into workflows. So it's not just about giving you a box where you can take the box and you can do one thing or one particular part of an application. You can actually do a lot more than that. So we are developing workflows, especially when it comes to complex matrices. And what would that one complex matrix would be other than the biological matrix? That is called FFPE. So formalin fixed paraffin embedded. Now, you may not be using, but are you all aware of what FFPE is? Right? So you have the tissue in the wax block. Now, it is amazing that such a technology exists, that someone in upstate New York has a tumor that's been operated and a slice of that gets into the wax block, and it can go to Los Angeles or in a different country without losing its integrity. The challenge lies in how do you get that tissue out of that wax block? So now imagine, this is not like a frozen food that you can buy from grocery store that's packed in ice, right? That ice will melt away. It's wax. It can go through hot, humid conditions without losing anything, right? So how do you remove that paraffin? How do you remove that wax? Typically, what people do is they use something called xylene, or there are mineral oil-based kits, reagents available. So you start washing it. And over a period of time, it's like, you have a stain in your shirt and, you're washing it unless and until that stain goes away. But with so many washes, the shirt is also faded and, frayed, if at all. So that also affects the tissues. With all those organic chemicals, it's also going to affect the tissue. And with that, it affects your analytes. So you do not get as much of a good quality of DNA, RNA, or proteins, whatever you're trying to use. Because the proteomics people, if you look at the systems biology, the omics people, metabolomics people will say, we are the king, right? Because we understand the metabolomic profile that is causing the cancer. Proteomics will say, I see the biomarkers. You guys don't, because at the end of the day, you have only few metabolites. I have specific protein biomarker. And then the transcriptomics will say, we look at the RNA, DNA, and the proteins. And genomics people will say DNA, RNA. Everybody's right. But one thing that is common across all of them, no matter what they're doing, is what? It's the quality of sample. You have to have good quality of samples in order to, as probably all of you are aware of the term or phrase, junk in equals junk out. So you need to have good quality sample coming in to get good quality data. So extraction is a huge issue. Xylene is a known carcinogenic. And for those of you who have come through chemistry labs or are still working in chemistry labs, you'll laugh at this and say, big deal. What is not a carcinogenic in the chemistry lab? Everything is carcinogenic. That's why we live in the hood. But this is known as a much more corrosive, harmful chemical, which is why you can't put this on a regular bench at all. So exposure is very, very difficult. And the other thing is yield because if you are washing it so many times, your yield is going to be compromised of the analytes that you are targeting. And last but not the least, imagine you're doing this with one tissue sample, one block. What if you have 96 of those, or hundreds and thousands of them? And those are not uncommon. Even 30, even 20 of those, you have how much time would you be spending? So AFA is a technology which does not require any chemicals whatsoever in emulsifying wax and exposing the tissue out. So this energy is not just for matrix, but also for extracting the matrix first. So now imagine what I said before and what I'm saying now. You have your FFPE or your scroll of wax with the tissue in it. You put it in a tube. AFA first switches on and gets the wax removed. Then it disrupts the tissue, gets your analyte out, and then it separates the analytes, DNA and RNA, and then it shears the DNA, if needed, RNA. So in one workflow, you're getting pure DNA, pure RNA. I'll come to the proteins in a bit. So we have this whole protocol now and all of that we provide, and it's also compatible with automation. So any of the liquid handlers will work if somebody is interested in working with automation. The efficiency of the system, of the technology. So if you go through the list, just look in this column. If you see that between all the other kits and reagents that are used today for FFPE, the amount of scroll that's needed is significantly higher than what we are asking for. And why is that? Because the yield is not guaranteed. One of the main reasons. That's why you start with a higher amount so that you have sufficient quantity that you can work with. And you don't necessarily need to worry about that. So no contamination, no temperature degradation, and the least amount of input volume that you will need to get to enough amount of DNA and RNA. So this is the workflow. And once again, there's no quiz. So you don't have to worry about, and we have this available in the brochure also on our website. So you can go through that. And I'm just going to say once again what I mentioned a few minutes ago or a few seconds ago about what this workflow is. And that workflow is primarily focusing on looking into, you take your 96 tubes or samples. So all the calculations you are seeing over here is based on a full 96 rack. It's significantly less if you're working with fewer samples, of course. So you take your samples in a 96 rack and you add some buffer and you wait for 5 minutes and then you switch on the AFA. Gone. First step, what's gone? The paraffin moved. So it emulsifies your exposed the tissue. And then you incubate it and you subject it to AFA again. Now you have disrupted the tissue. Out comes the DNA and RNA. And then we again provide some cross-linking buffer where you will now see a pellet in the tube and a supernatant. So this pellet is the place where all your DNA is. And supernatant is where your RNA is. It's easy to separate a pellet from a supernatant, right? You separate that, RNA is on the left side, and you go through incubation of, do add the binding buffer and all those things, you get pure RNA. To the right is the DNA, which is a pellet. And of course, when it comes to pellet, you have to first do what? You have to reconstitute it in the buffer. And then from there on, you do follow a similar protocol and you get your DNA. So typically, one sample or a few samples, it takes 96 samples for a lab to go through the process that I detailed on and what they typically use today. It goes into 15, 16 hours of sample preparation. You get the whole rack in 5.5 hours with pure DNA and pure RNA. That's what you're getting. So at least about 3 times faster for all 96 samples. Now imagine in a research environment, you may not have 96 samples. So you work with fewer samples and you are done faster also. Now, if you're going to ask me that can you divide 535 minutes by 96 and get a number like 5 or 6? So is it 6 minutes per sample? No, it isn't, because you need a minimum incubation time in certain cases. So it's going to be more than that, but it is definitely going to be significantly less, at least 3 times less, factor of 3 compared to what people are doing otherwise. So some data that I'm going to say, once again, we provide the full workflow with our buffer chemistries, everything included. We provide the consumables, we provide the instrument, and this instrument, you will also see a brochure back there. And then if you need automation, we provide that. So here are some examples, of course. I cannot not show examples, right, or data. So over here, we've taken 4 different sarcoma tissue types. It's brain, breast, colon, and lung. You see the amount of tissues. So that's how the blocks look like, the wax blocks. You see the amount of tissues are significantly varying. Must be some intelligent person with the brain. It's not mine. I wish I had that. Not the cancer. So all of those things are there. So as I mentioned, when you have that block, what do? You use a knife and you take a scroll out of it. That has got a lot of wax and a part of the tissue, which is why people do multiple scrolls so that they have more tissues in it. We are saying we probably don't need as much. And we start with cutting using a rotary microtome at room temperature. And then we get a 10-micron scroll, as I mentioned. We then process it using the workflow that I showed in the previous slides. So how's the data looking like? So RNA data, we look into multiple factors. First and foremost, it's the RNA, the yield and purity of the RNA. It's very vulnerable as an analyte. For those of you who work with RNA, you would know it is an extremely vulnerable analyte. It's unstable. So you look into the 260-280 ratios, the 260-230 ratios. And over here in the data, you see two blue bars, or one grayish blue, one dark blue, right? These two. What do they stand for? Both are our workflows. One is the automated kit, where we are using an automated platform. And the other one is where you can use the same kit, but some different protocols manually. So if you don't have as many samples, you don't have a volume requirement, you can use less samples or a less number of tubes. So that's the Auto 96 manual kit. They perform similarly. So we are looking at better yield and better quality. And if I move over to the amplifiability of the RNA, you are also looking at better quality of the FRQ values and also better TV200 scores. The one in the yellow is actually something we are using a competitor. Which competitor? I'll come to it in a second. Not showing it on our slide, but using a publication that came out recently. So similarly, we get the same thing when it comes to DNA. You get better yields compared to better quality yield. So it's not just better DNA, but the DNA that you actually can work with. So better quality DNA as an yield, and you get the same nanodrop ratios. Same thing, the kappa QC scores, and the molecular weight that we do by smear analysis, which talks about the amplifiability of the DNA and the molecular weight. So that is just the total DNA and RNA. The next thing is that you share the DNA, right? Before you amplify it, you share the DNA. And we are using over here some of the competition kits as well. So while right up front, they all look same in terms of profile, there's less reproducibility here compared to here. So that's where the quality of data is, and that's where the value is coming from. We can go over, this is uterine tissues, kidney tissues, you get the same thing, better reproducibility with AFA and less reproducibility while using competitor kits. So the target was to get to 175 base pair DNA. You get not just that, but not just better yield, but better reproducibility, which is what you need. Imagine you are working with a sample which is going to, your analysis is going to tell a person whether he or she has cancer or not. It's deciding the life of a person. So how important is it to actually understand how confident your data should be? So that's the degree of difference that can make or break a life, right? Same thing, you can show the colon on uterus. So this is the publication that came out of a Finnish group. This came out in 2018, where they were using an early adoption version of our kit. So they were comparing, their main goal was to do high throughput screening. So they get lots and lots of tissues from different sources, and they do this high throughput screening. It's a central laboratory. And they isolate the DNA and RNA from FFPE, and then they look into their holistic quality. So they compared all available kits, reagents that are available, and look at over here. So if you just do a basic math, there are some common, these are all different sarcoma tissues that they are getting. So different sources from different organ types. And if you look at it, the number of genes that they are identifying that can actually help them. Whenever you have such situations, the more contact points or confirmation points you have, the better your result is, right? So you are getting much better data when they are using this kit. Same thing over here, you get the same results over here. Some are comparable and some are better. But at the end of the day, they are also very reproducible. So there's more confidence and reliability associated with this. So that's DNA and RNA. And now I move to my favorite topic. And most of you are not working with proteins. So I understand. Your least favorite, my most favorite. I dealt with your partiality until now. Give me a few minutes to work with proteins. So the best thing about proteins is that they are all very different. The worst thing about proteins, they're all very different, right? So how many different kind of subunits does a DNA or RNA have? 4. How many subunits does protein have? 20, right? And they come and join those amino acid sequences that decide how a protein is different, and every single protein is different. I mean, our body has, what, 200,000 different kinds of proteins in the cell. And so you're looking at truly, when you're trying to find a protein biomarker, I call it, I came up with the phrase 12 years ago, that you're looking for the needle in a needle stack. It's not the haystack anymore. You're looking for a specific protein in an ocean of proteins when you're trying to find a biomarker. Now comes the problem is when you have a tissue, you disrupt it, very good. You get your proteins out. What do you do with it? Your first level of proteins is that all that IgG, albumin, which are abundant proteins. By the way, clandestine information, they are not needed in terms of your research. You are not interested most of the times in looking at the IgG or the albumins. They're the most abundant. As Murphy's law, right? Whatever we look for is not available in abundance. We are looking for those trace protein biomarkers that are going to tell us the course of a disease. Most cases. There are lots of scenarios where IgG is looked into. So what people do in scenarios like that, and he can do a much better job in explaining protein depletion, I'll try, that you have an ocean of proteins that has come out of a tissue. And now you have got all those proteins and you want to separate the abundant proteins from the non-abundant ones. So you add a bead, which is part of an immunodepletion process, where your less abundant proteins bind to the bead and you clear out the rest. You take the supernet and forget about it. So now you've got the beads. Now you have to wash those proteins, or these are humongous proteins. And what you are doing at the tail end, how many of you are using LC-MS? Anybody using mass spectrometry here? One. Okay. That's fine. That's fine. You're all allowed a mistake. So I'm just kidding. I've worked with mass spec since 1997 until 2021. So I'm a bit partial to that technology. But typically, mass specs are very good, very specific, very selective, right? But they can't handle a big molecule. It's not your sequencers, not your PCRs. You cannot amplify a protein, by the way. So when you work with mass specs, you have to now do, you have got a gigantic protein. The molecular weight is what, 150 kD, if not higher. And your mass spec can only see this much at a time. Your mass spec can see 1,000, 1,200 Dalton at a time, and not more than that. What do you do with that big protein? You cut it into smaller pieces. That's the digestion part. And then when you digest it, imagine if you had an ocean of proteins bound to the beads and not the most abundant ones. Now you have this ocean of proteins chopped, and now you have multiple oceans of peptides. And you're looking at specific peptides pertaining to specific proteins from there. How difficult is that? It's all about purification, preparation, so that your mass spec can get to the point. We can provide all that in one plate. So you don't have to change the plate. All of that can happen in the same plate. And so you disrupt the tissue or you homogenize the cell lyses. And then you look into it and it's like, oh my God, I have got so many proteins floating around. You do the immunodepletion. AFA assists in the binding rate of trace proteins onto the beads so that you can remove your abundant proteins, which is not what your goal is, easily. And from there on, you digest the proteins on the beads in presence of AFA. And you get the peptides, and then you purify them in presence of AFA so that you can get to the final point. Whichever technology you're doing, if you're using ELISA, if you're using some other form of technology, you can use, it doesn't matter what your anti-detection technology is, you are getting good quality proteins at the end. So with Matthias Mann, we have got a publication out in 2020. He calls it APAC. It doesn't stand for Asia Pacific. It stands for AFA-assisted protein aggregation capture, right? So what he has done is basically, he's worked with fresh frozen tissues, FFPE tissues, all of those kinds of things. So he takes those, does that whole thing, the entire protein purification, isolation, and digestion workflow on an automated platform, which is the Agilent Assay Map Bravo. And he then puts it in the mass spec and he gets his data. He does it in an automated fashion. And what he's using over here is the AFA, which is happening in the background. So it's very complex, but once you get into a rhythm, it's actually not that bad because you let the system run the course for most part of the times. This is a publication that's still not out, but it was a poster that was presented by Jessica Chapman from Memorial Sloan Kettering in New York. She presented it at ASMS this year. So she's looking at different kinds of tissues. She's also looking at IgG proteins and IgA proteins. And she has this whole workflow where she's using AFA at every stage. So I'm not going to go over every single details that are mentioned over here, but from those tissues, what she's highlighting over here is when you keep the AFA on, the whole digestion protocol for proteins, it takes 16 hours at 37 degrees normally. She's digesting it for 3 hours with AFA on, and that's more than enough for her. Her binding efficiency is also much better. So it's significantly less amount of time. She's seeing equivalent or more proteins identified in her mass spec assays. What does that mean? In half or less than that of time, you are actually seeing much better protein identified. In there lies your biomarkers. And with those biomarkers, you can run and figure out that these are the common biomarkers in this types of cancer. We also have several protein extraction protocols using different instruments, where here in this particular case, we are using a different technology. We are using a fluorometry principle to look at proteins, but all of those are possible and different temperature conditions. So everything is assisted by AFA with higher yield and higher quality of proteins. We have multiple workflows, as I mentioned. We are heading towards that direction where we are working with workflows. And that makes a lot of difference because you don't necessarily get a part of a workflow as part of what you're doing, but you get the whole thing optimized for you. So in summary, what I'm trying to achieve today is that in addition to all of the tissues and matrices that you're working with, which can work very well, and we have got some AFA users over here who can also tell you about their experience with the AFA instrument downstairs. But in addition to that, there are some more complex matrices that are coming up, such as FFPE, and it is very challenging to work with them, especially if your number of samples go up. And that's where we come in and we are providing this holistic, comprehensive workflow that can help you not only remove the paraffin, but work with better quality analytes and higher yield, DNA, RNA, proteins, all alike. So we have a host of instruments in our portfolio. This is the one that we are primarily using for our 96 well plate workflow. These are also capable of handling 96. These can handle 8 samples at a time. We can make our FFPE workflow on multiple platforms. As of now, it's working on this particular workflow that we are talking about. It works on these instruments. We are getting towards including them. We provide our consumables and reagents also part of it. And we have offices and centers all over the world. So that's about it from my end. We have about 15 minutes left. I may have sped up somewhere in the middle. So now I actually thank you so much for your time. Hope the lunch was good. Hope the talk was okay, maybe not as good, but any questions? I appreciate your time. Thank you for making the time, and I would be more than happy to answer any questions that you may have. Thank you. So, any questions? Yours at times. People have reported, like the publication that I showed for DNA and RNA, some of the samples were 18 months old. So the way it happens is that it's a very convoluted process. The hospitals primarily get to the tissues first, right? Because they have the tissues. They put it in the black or in the wax block. And then the pathologists start taking scrolls. And for the biopharma companies like, the Novartis's, Pfizer's, Genentech's of this world, for their initial analysis for cancer drug, they have to buy those scrolls. The hospital biomarker research centers get some. And so, and it goes all over the place, right? So some of them are as long as 2 years, 3 years old. Yes. Different animals. No, we haven't had that. But I'll put a spin to it. We have looked at plant tissues. We have looked at some microbiomes as well. So yes, we can address them. At the end of the day, there shouldn't be an issue with that. Yeah, we shouldn't be an issue at all. I mean, with Johnson & Johnson, for a proteomic assay, we looked into skin tissues of dead people. So they are as hard as it gets. And with this technology, it homogenized those tissues, of course, at a higher power. But yes, it shouldn't be an issue. Any other questions? Yes. We have the Ultra Kit, which is a different chemistry that we have that can work with that, which works with one sample at a time. And yes, you can. I mean, those are also available. We first had the Ultra Kit that I mentioned, and then this is something that we launched this year. The chemistry, yeah, go ahead.